I will be explaining and modeling the propagation of an action potential down an axon of a neuron. Keep in mind throughout this video, propagation is just another word for conduction. I will be using both interchangeably throughout this video. Just remember, they mean the same thing. The action potential I will be talking about deals with the brain and helps make the central nervous system, otherwise known as CNS, and the peripheral nervous system, PNS, work. CNS deals with integrating and processing sensory data and motor commands inside. PNS involves all the neural tissue outside the central nervous system. Now that you know the locations of action potential I will be telling you about, next let me explain what action potential is. Action potential is the propagated changes in the transmembrane that, once initiated, affect an entire excitable membrane. It can be best explained as an impulse or a spike that occurs when neuron sends information down an axon away from the cell body. An axon is the long cytoplasmic process that makes action potential possible. It also carries out information to the cell. It is the long part in this picture. A neuron connects with the axon and is an electrically excitable cell that processes and transmits signals, which is at the beginning, as you can see in the picture. There are two types of ways action potential occurs down an axon of a neuron. They are called continuous conduction and saltatory conduction. Both types relay messages down an axon. For example, action potential could be visualized as a simple message from ear to ear, and the speed varies. It is just a matter of how fast it is relayed and what is helping speed up the process. Before going into detail about these kinds of propagations, let's talk about the differences. The differences can be seen in the membrane resistance, length constant, and conduction velocity, in addition to obvious different structures of the axon. Membrane resistance increases with saltatory propagation and decreases with continuous propagation. Length constant is the length potential that will travel, and conduction velocity deals with the speed of impulse. Saltatory conduction is fast and has high speed length potential and conduction velocity. Continuous propagation is the exact opposite and has much lower speeds of impulse. Now I will get into the structural similarities and differences of both kinds of propagation. Both have an axon and neuron, as you can see in the pictures. Major differences include the fact that saltatory propagation's axon contains myelin sheaths, oligodendrocytes, Schwann cells, and nodes of Revier. Myelin is of the myelin sheath and is responsible for speeding up nerve impulse. This is why saltatory propagation is faster than continuous. Continuous contains no myelin. Myelin sheath is a membranous wrapping that coats the axons of saltatory propagation, as seen in this picture. The myelin sheath is made up of two kinds of neuroglia, oligodendrocytes and Schwann cells. Neuroglia are cells of the nervous system that support and protect neurons. Oligodendrocytes help maintain cellular matter and create myelin sheath. Schwann cells are responsible for providing surrounding layers of the axon. The nodes of Revier are also known as the gaps that separate adjacent intermodes. Saltatory conduction can be described as node hopping. The local current in this kind of conduction creates graded depolarization. Any shift from resting potential toward a more positive one is known as depolarization. The charges change at the action potential site. The action potential occurs between the gaps of the nodes of Revier. Sodium ions begin to make their way into the inner membrane surface, and potassium ions begin to make their way out. These ions make their way in and out through voltage-gated ion channels, which will be explained and pictured later on in this video. With continuous conduction, action potential spreads across the membrane in a series of small steps because there is clearly no myelin to speed up the process. The axons are unmyelinated. At the peak of action potential, the transmembrane potential turns positive rather than negative. Depolarization helps move sodium ions and keep the process going like a chain reaction. How do sodium and potassium ions make their way in and out of the membrane? The answer is voltage-gated ion channels. The voltage-gated sodium channel depolarizes action potential to allow an opening for ions. Once the sodium channel is deactivated, potassium moves out of the cytosol, changing the transmembrane potential back towards resting levels.